Hi everyone, welcome back to VRC Smart YouTube channel. So today I am with class 12 biology chapter 5 principles of inheritance and variation. So you all know that we have made previous 4 parts and the link is provided in the description for all the 4 parts. You can go and check it out. So the 4 parts are going to be really important for you. So go and watch it. So now today we are going to see part 5 of this chapter, principles of inheritance and variations. So in this part 5 video, we are going to see about the linkage and recombination, polygenic inheritance, pleiotropy. Okay, these 3 concepts we are going to cover in this part 5 video. So if you are new to our channel, please subscribe to our channel and click the bell button for notification. If you like this video, kindly like it. Come on, let's move into the video. So, in the previous session, that is in the previous class, we have discussed about various things like incomplete dominance, codominance, right? So here I am going to talk about an example, okay? So take a pea plant. Okay, we all know what is a pea plant, right? Pisum sativum is the biological name of it, right? In pea plant, what I'm going to do is I'm going to take about the starch synthesis. I'm going to talk about the starch synthesis. So the pea plant, one of the gene is responsible for the synthesis of starch. So now what I'm going to say is the gene that is responsible for the synthesis of the starch, let it be B, and its allele B, small b and capital B are the 2 alleles. These are the genes that are responsible for the synthesis of starch. So if the starch synthesis is going to be capital B, then it is a dominant character. You all know that, we already know that capital letter represents the dominant character and small letter represents the recessive character. So capital B is dominant and small b is nothing but recessive character, right? Dominant character, recessive character. Now what happens is, see, this is dominant character and this is what? Homozygous, right? Homozygous. This is also homozygous. Now tell me, how will the plant look when it is having, how will be the seed when it is having capital B, capital B? The seed will be having more starch synthesis. Okay, the seed will be having more starch synthesis, that is, it will be more efficient in starch synthesis. It will be more efficient in starch synthesis. Here it will be less efficient in starch synthesis. Okay. So similarly, if you see capital B, as it is dominant, how the seed will look like here? The seed will be very much large. The seed will be very much large. Here the seed will be large and round in shape. Round, large and round. Starch grains. You will get large and round starch grains. Here you will get small and wrinkle starch grains. Okay, so these are the things you have to remember it very well. Okay, so now what I'm going to take is, I'm going to take the capital B, small b. It is also one of the character, right? We saw about the completely dominant character, completely recessive character. It is capital B, small b. Usually what we studied in our Mendel's law of dominance, a dominant character will be expressed like that we studied. If the dominant character have to be expressed, what happens is, you have capital B, small b, right? If the dominant character has to be expressed, you know that large and round starch grains you will be getting. But here the case, you will not be getting the dominant character. Here intermediate intermediate character only you will be observing here. Intermediate character means the seed can be medium sized, it will not be round as well as it will not be wrinkled. In some intermediate shape you can get it. In case of size, if you take, in case of size you can get the intermediate size. So this, if you consider the size of the pea plant, then it represents the what? Incomplete dominance. It represents the incomplete incomplete dominance, right? Incomplete dominance, this is one of the important point that you have to remember here. Okay, so come on, let us move to the next topic. Next we are going to see about the linkage and recombination. Okay, so what is linkage and recombination? Let us see now. Okay, so listen here carefully. Morgan carried out several dihybrid crosses. Okay. Morgan, already we studied about Morgan. He only described about Drosophila melanogaster, right? He took that fruit flies and he had observed it. He had done an experiment on it. So, for the chromosomal theory of inheritance, we already studied this one, right? So, he took it and he had done some experiments, and the reason for selecting this fruit flies, that also we studied in our previous class. Okay, so Morgan carried out several dihybrid crosses. Now he took that Drosophila and he tried out several dihybrid crosses on that. Okay, to study the genes that were sex linked. Okay, how the genes are linked with each other. Okay, so first he took the yellow bodied and white eyed females. So yellow body and white eyed females here are recessive character, and brown bodied red males he took, that is a dominant character here. That is the dominant character. Okay, so when he crossed that yellow body and white-eyed females, there are two characters. So you say it as dihybrid cross. This is one character, body type and eye type. Right? This is the second character. There are two characters, so it is dihybrid cross. So he got the F1 progenies. So first, when he crossed, for example, you take the yellow body and white-eyed females, right? So yellow body and white-eyed females is crossed with brown body and red-eyed male. So when these two are crossed together, you are obtaining the F1 generation. Okay, you will be getting Y, right? So Y and W will be splitting here also. Y and W will be splitting. So, and your F1 generation, you will be putting it in the Punnett square. You will be getting the F1 generation in this case. After getting the F1 generation, he intercrossed the F1 offspring. That is, for example, this is my F1 generation. He crossed the F1 generation together. He found that two genes did not segregate independently. So, they did not segregate independently. Two genes were not independent of each other. In case of law of independent assortment, we studied that when you cross two different characters, one character will not be dependent upon the another character, right? So, but here they are not independent, they are dependent upon each other. Resulted in deviation from normal hybrid ratio in F2 generation, because appearance of the parental combination were higher than the non-parental characters. So what they are saying is, see this, they are saying that you have the ratio 9:3:1. This ratio is obtained in the normal dihybrid ratio, but it will not be obtained here because you can able to say they are not segregated independently. Okay, don't confuse yourself. While reading the diagram, you can able to understand this one easily. Okay, so now let us try to understand what is the meaning of recombination and linkage. Okay, so if you just see recombination, it is a generation. Okay, it is a generation of non-parental gene combination. When you combine this two uh, this Drosophila, no, it is when it is combined, no, you are getting the F1 generation as well as F2 generation. In your F2 generation, you can able to observe some non-parental character. That is the characters that is not present in the parent. That type of character you can observe here. That character is called as, if you get that character is called as recombination here. Clear? Okay. Linkage. What is linkage? Physical association of two or more genes. So, physical association of two or more genes on a chromosome, which shows a tendency of inherit together. They do not show independent of assortment. So, we already studied, right? They will not be independent of each other. They will be dependent of each other. We already saw this in our previous slide. Yes? So, see here, you can able to clearly observe here cross A, double recessive. So recessive character, you are taking yellow body and white eyed females. That is, this is the combination, and you are crossing it with brown body, red eyed male. Wild type means dominant type. Okay, so recessive type and dominant type are crossed. Then double recessive, so white eyed. This is cross B. So cross, two types of cross we are doing, cross A and cross B. So both the crosses I will be showing you. So cross B, what we are doing means we are taking another two different characters, white eyed and miniature winged. So first cross A, we are considering the body color and eye color. These are the two characters we are considering in cross A. In cross B, we are considering the another two characters, that is going to be, one is the eye color and the shape of the wings. These are the two characters we are considering in cross B. Okay, so here we are taking the miniature winged means small winged females, red eyed male and large winged male. So here also male is dominant and female is recessive. Clear? Listen here. So now we are going to see the diagrammatic representation of it, and we are going to study it using the diagram. So you can have a better clarity when we study it using the diagram. So let us do it. See, this is cross A. So first we are having the parent. You know that this is the female, right? You know yellow body it is having, white color eye it is having. So it is having the X chromosome. Actually, what happened is, Morgan, no, while studying, no, they found that the X chromosome is going to contain the genes. So X chromosome will contain the genes responsible for eye color, that and all. But Y chromosome here will not contain. This is X chromosome, this is Y chromosome. So in case of male, so Y chromosome is not going to contain any genes. Okay, it is not going to contain genes that are responsible for any characters. Okay, so see here. So now you are going to see here. Here you have two XX chromosomes. So this XX chromosomes will fuse with this XY chromosome. While it is fusing, no, you will get your F1 generation as, see, this is your female in your F1 generation. This is a male in your F1 generation. Here the male is representing the female character, right? Yellow, white. Here the male is representing the male child. You can call it as offspring or baby. The male offspring is representing the female and the female offspring is representing the male characters. This is very, very important to remember. In the F1 generation, the female offspring is having the characters of the male father, the male offspring is having the characters of the female mother. Clear? Now, in the next, what is happening is, next he is crossing the same F1 progeny he is crossing. So this F1 progeny he is crossing, so that he is getting the F2 generation. In the F2 generation, what happens is he is getting the this types he is getting. You can able to see that wild type, yellow white, wild type. For four he is getting here, and white, yellow, he is getting this also. If you are able to observe here, wild type, yellow, white, these are the characters that are obtained from the parent, or we can call it as parental type, right? So you can able to see that the parent is also having the same type. So these are the parental type. So here you can able to see that the type which are not observed in parents, white is not there in parent. Maybe one character it may be present, but it is not completely there in parents. Yellow is not completely there in parents, right? So here if you just look after means it is not completely there. That means it is a new character that is formed. If a new character is formed, already we studied new character form means we call it as recombinant. So these are the recombinants and these are the parental type. Here in cross A we have parental type as 98.7% and recombinant types we have 1.3%. Okay, which is comparatively very low. The recombinant type is comparatively very low compared to the parental type. Parental type is high, but recombinant type is very low. Okay, so this is cross B. So if you see means cross B is having white miniature wing and wild type. The wild type is what? Long wings with red eye, right? This is male character. Here also you can able to observe this is the X chromosome, Y chromosome. This is X chromosome, X chromosome. So Y chromosome is not having any genes, right? So if you just observe in the second case, here are your F1 generation produced. So in your F1 generation, if you look after what is happening in your F1 generation, here also you have the same progenies observed. See, here also you can able to observe that the female daughter is showing the character of the male father and the male son is showing the character of the female mother. Okay. So now you have the gametes. After formation of gametes, you can able to see wild type, white miniature, wild type, white miniature. So these are the parental types, and miniature, white. These are the recombinant types. So when you are comparatively observing here the parental type and recombinant type, it is much higher, right? So compared to 1.3 percentage, 37.2 percentage is much higher. So with the help of the parental type and recombinant type, so we are going to say one thing based on this. So you can say that the cross A, is tightly linked. Cross A is tightly linked. And you can say the cross B is loosely linked. Okay, so you can say the cross A is tightly linked and you can say cross B is loosely linked. Clear everybody? Got that? Yes. So this is all about this diagram you have to remember. Clear? Now let us see what Morgan had found when he crossed. Okay, so Morgan crossed and found that two genes did not segregate independently of each other and F2 deviated from the ratio 9:3:1. So they did not segregate independently of each other. Okay, and they are deviated from the ratio 9:3:3:1 ratio. The F2 generation. Genes were located only on the X chromosome. We are able to observe Y chromosome did not contain any genes. Only X chromosome contains genes. When two genes were situated on the same chromosome, the proportion of the parental gene combination was much higher than the non-parent type due to linkage. So because of the linkage, you can able to observe that the parental type is high. Parental type is high. The non-parental type is comparatively low, right? So you can able to observe in the first case 98.7%, in the second case also you were able to observe 68.7 percentage or 68.6 percentage. You can able to observe that, right? So, but the non-parental percentage was comparatively very much low. Gene for white and yellow were tightly linked. Okay, and showed 1.3% of the new recombinant, while white miniature ring showed 37.2 recombination, loosely linked. Already we studied, right? Tightly linked genes show low recombination, loosely linked gene high recombination. So here you can able to see that tightly linked gene low recombination. So here low recombination, right? Low recombination percentage. So you had how much? You had tightly linked genes. Okay, so loosely linked genes, here you have high percentage of recombinant, so you have loosely linked genes. Now clear? Yes. Coming to genetic mapping, this is really important. Okay, Alfred Suterman used recombinant frequency. He used recombinant frequency between gene pairs to measure the distance between genes mapped in their position on the chromosome. He used to map the genes with the position of their chromosomes. So where it is, for example, he say here A gene is present, here B gene is present, here C gene is present, like that. So you use the recombinant frequency. How to find this recombinant frequency? This recombinant frequency, how to find is recombinant frequency is equal to number of recombinants. Number of recombinants, number of recombinants divided by total number of offspring, total number of offspring into 100. So this is your formula for recombination frequency. Clear? So genetic maps are used as a starting point in the sequencing genome. Okay, it was done in Human Genome Project. This is really important to remember. It was done in Human Genome Project. Okay, right. So now let us move to the next. So now coming to the next one. So it is a very easy topic. Okay. Polygenic inheritance. What is polygenic inheritance? Polygenic inheritance. So traits controlled by three or more genes is called as polygenic inheritance. So now, for example, I am taking only the human skin color. Okay, it's an example of human skin color. You can able to understand it really easier. So A, B, C are responsible for the dark color. So your A, B and C are dominant character. What are the alleles of A, B, C? Small a, small b and small c are the alleles. Alleles of your A, B, C. Okay, they are responsible for the light color. Okay, so if your formation of your character is capital A, capital B, capital B, capital C, if the formation of the genotype is this, what will be your phenotype? The person will be the darkest. The person will be the darkest of all. If the your genetic code is something like this, small a, small b, small c, what will be the phenotype of the person? They will be the lightest. They will be having the lightest color. Okay, color. So apart from these darkest and lightest, intermediate genotypes are also available, right? So in between genotypes are also available. In between phenotypes are also available. Okay, intermediate phenotypes you can able to observe, right? You can able to observe the person who is not too dark, not too light. Okay, you can able to observe the person. There are various different types of skin types you have. Okay, not only darkest skin, not only lightest skin, various different types of skin types you have. So you can't determine it. So you are having three or more traits is controlling a gene. Gene is nothing but human skin color is a gene. So more than three trait is controlling the particular thing, that is called as polygenic inheritance. Clear everybody? Okay. Moving to the next. Next is pleiotropy. Okay, what is this pleiotropy? Let us try to understand it quickly. Okay, pleiotropy. Now, here you see means single gene responsible for multiple phenotype. You are having only one gene, but this one gene is responsible for more than one phenotype. Okay, more than one, more than one or two, more than one phenotype. So let us try to understand it. So example is phenylketonuria disease. It is an example. How this disease is caused? This is really important. This disease is caused by mutation in genetic code. Mutation means some differences occurring in the genetic code. Okay, for the enzyme phenylalanine hydroxylase, single gene mutation. So this is the name of the enzyme. This enzyme is getting muted. Okay, mutation is occurring in this enzyme. So single gene mutation is taking place. So what happens is, so when this mutation occurs, you can able to observe this pleiotropy. So here it causes the the phenotypic expressions are, here you can able to see the people having mental retardation, reduction in hair will be getting reduced, skin pigmentation they can able to have. So these are some of the phenotypic expression that the person can have. Clear? This is called as pleiotropy. Clear? Okay. So that's all for today's video. Thank you so much for watching this video, and we will be coming up with future parts based on this chapter. If you like our video, kindly like this video and share it with your friends, and subscribe to our channel and click the bell button for notification. Follow our channel. Thank you so much for watching this video.